Here we present a simple kernel screening assay to analyze Aspergillus flavus growth and aflatoxin production in maize kernels expressing an antifungal protein. Using a green fluorescent protein expressing Aspergillus flavus strain, we monitor the infection and spread of the fungus in mature kernels in real time. This simple assay provides a reliable means of evaluating the maize kernel's ability to resist Aspergillus flavus infection and aflatoxin production. Results from this lab assay performed under controlled conditions on mature kernels correlates well with results from infection under field conditions. Demonstrating the procedure will be Dr. Rajtilak Majumdar, a postdoc from our laboratory, along with Christine Sickler and David Ambrosio, biological science technicians also from our lab.
To begin, glue 22 mm snap caps in a petri dish to create KSA caps. Let the glue dry for 48 hours before using the caps. In a biological safety cabinet, spray a square bioassay tray with 70% ethanol in water. Let the tray air dry. Add sterile chromatography paper to the dry tray. Spray 9 of the KSA caps with 70% ethanol and let them air dry. Then, place the caps in the bioassay tray. For each transgenic maize line being tested, select